let's move on to the next section of our chat, where we have a special guest who today is Dr. James Fleshman. He's got a phenomenal biography of achievements. He's been the president of ASCAS, been the president of the American Board of Colorectal Surgery, president of ASCAS Research Foundation. But what I found today, which was most interesting, is that he wears cowboy boots on Fridays. But the aim of this discussion is to focus on the Z6051 trial, where he was a key player, but not so much to discuss the significance of the trial. We all know it's a seminal paper published in JAMA, but more to sort of pick his brain about the mechanics of undertaking this trial and seeing it to completion. And so my first question is, when and how did you decide on running this trial, and how many years did it take from conception to completion?
Well, thanks, Vlad. First of all, thanks for including me in this. And I want to congratulate Dr. Daly and Dr. Parikh on some excellent presentations. As soon as the colon trial, the cost study trial was over, we all started talking about rectal cancer. What are we going to do about rectal cancer? We had to decide whether rectal cancer was different from colon cancer. And the discussion was around, is it equivalent in terms of ease of minimally invasive approach? And we all agreed that it wasn't. There was not a consensus that minimally invasive surgery for rectal cancer was the next step. So we felt like we ought to at least put it to test. So to Heidi Nelson's credit, she allowed us to start a protocol. And when she became the head of the American College of Surgeons Oncology Group after Dr. Wells stepped down, she told us to go ahead and put that into gear. So in 2001, we started writing our protocol. David Dietz took it to the GI committee several times for me when I couldn't make it, but we waited 8 years from the time of starting the protocol to the time it was finally approved and we could move forward. The writing of a protocol really is not easy, especially when it's going to be attached to a $3 million request to the NIH. So Heidi gave us Dr. Anne-Marie Buller, who was in Chicago at the time and was an outstanding asset. She and I went back and forth on a daily basis, writing this protocol, honing it. I had an executive committee that was really looking at every change we made. Every meeting that we had we could all gather together, the ASCRS, the ACS, the SAGES, SSAT, Society of Surgical Oncology. There was always a group of people there who wanted to sit down and talk about putting that protocol together. And so it was clearly a joint effort. I had an executive committee that worked with me. Just for example, John Monson, John Marks, Steve Wexner, Tom Reed, Peter Marcello, Mark Whiteford. It was a joint effort. Everybody was sold that we needed to get the right answer for this question. Had I tried to do this on my own or tried to establish a protocol and put it through without the backing of the entire Society of Colorectal Surgery, it would have been a mistake. And the reason we even got the trial done was because of the commitment of the colorectal surgeons in our country. And we designed the trial so that it could be duplicated elsewhere. So Andy Stevenson and Gop Bonger and the Korean group all took it and put it together. And we're going to have a meta-analysis soon of a metadata analysis soon to see if our conclusions are correct. But that's how a trial worked. best for us and how I think we can all feel comfortable with the outcome.
Thank you. That's a very, I didn't appreciate how, I mean, I know it took a long time. I didn't appreciate how long. You mentioned that you sort of had a $3 million budget. At what point does one decide on a budget for a study? And, how do you keep within budget? Not only for something this massive, but for smaller studies that people might want to do, smaller RCTs.
Well, it starts like any clinical trial, prospective clinical trial should start, which is calculating your sample size. And I know you've got some questions about how we did that, but if you know that this is going to be a capitated trial so that each patient is going to be paid for with a certain amount of money, you have to be able to meet that. And you can calculate the needs of a trial based on the amount of data that's going to be collected, the number of times the patient's going to be seen, the number of people that will be involved in caring for that patient, and then the background noise that goes into statistics and data cleaning and a data safety management board and travel for the study group back and forth. It adds up. And I was very fortunate that CTEP, which is the Clinical Trials Evaluation Program, was interested in looking at our technical trial as the next, I guess, the next stage of doing prospective trials. And I tell you, Dr. Nelson is an amazing human being, but she's also a genius in my opinion, because she figured out a way of getting this through the NIH process in such a way that they really wanted to do this trial. So I wasn't having to knock down any doors to get things done. It was there, it was ready. And she really is due a lot of gratitude and a lot of respect and congratulations for what she did for us to get this done.
Okay, thank you. That's interesting. Focusing on the methodology of the study, it is a non-inferiority trial, meaning, obviously, but laparoscopic rectal resection being non-inferior to open. How did you settle on this compared to, say, an equivalence or a superiority trial? And specifically, I asked this, there's an interesting publication by Nancy Baxter's team in JAMA in about 2018, talking about interpreting non-inferiority trials, and they mentioned this one and also the Stevenson-Ellicott trial, and they demonstrated that the results were often misinterpreted in other papers, of which they quoted 71, and they described them as being quite ambiguous in their language. So what do you think about the methodology?
So Dan Sargent was the chief statistician for the American College of Surgeons Oncology Group and later the Alliance, GI Alliance. No, just the Alliance. And the idea is that if you wanted to do an equivalence trial, you would have to have close to 3,000 to 4,000 patients. If you do a superiority trial and you get no result, then you still don't know whether or not it's equivalent or whether you just failed to pick the right process. So a non-inferiority trial was based on the fact that if you look at a lot of the medical oncology studies, they are banking their conclusion on a 6% difference between the effective group and the ineffective group. 6%. Now, if you think about that, that's not a lot. And they're staking people's lives on that all the time. So a non-inferiority trial actually takes that 6% and says, if you can't get within that 6% and be close enough into the study group, well, then you really need to say that there's no difference and maybe there's no benefit. And so that's how the non-inferiority trial came along. Instead of 3,000 patients, we could do it with 800. And we could get it done in a period of time that was reasonable for us to be able to report it and it not be completely, irrelevant, which it almost was, because robot came out right at the end of it and people were already making the switch to robotic proctectomy, and we still didn't have our data out for the laparoscopic approach.
Maybe it's difficult for you to answer this, but if I'm not mistaken, the a la carte trial, they comment on a non-inferiority difference, meaning lab being worse than open, was 8% compared to yours being 6%. How does that work to a simple-minded person like me? Because it's the same operation.
Andy calculated what they felt like was a reasonable difference that they could hang their hat on, and I think it's the Australian GI tumor study group said that they wouldn't do it if it wasn't 8%. So he said, fine, 8%, whatever. The 6% was something that the NIH, the NCI, and every cooperative study group had been using in the United States for a long time. So there were no raised eyebrows when we suggested that. So if we said, if the open group could get 90% complete TME and complete distal margins and all the lymph nodes, then if you could get at least 84% with the laparoscopic approach, then it would be within the range of saying that there was not a lot of difference. And you could call it almost, you could call it a non-inferiority trial. So the laparoscopic arm wouldn't be inferior to the open group.
Just taking you up on the distal margin point, I think in the trial, the negative distal margin is greater than 98%. For me, I always worry about having a positive distal margin, and thankfully so far, the only times that happened, I've woken up in a cold sweat. And so it's never actually, never actually been on the pathology report per se. But how do you think, can we actually get to 100? And it seems to me like it's a preventable issue.
It's a preventable issue if everyone gets an abdominal perineal resection. Think about it that way. So reconstruction has made us compromise on saying that we want a 5-centimeter distal margin for every case that we do. During that time period, as we were trying to decide what to do with this distal margin issue, we actually noticed that there were people who were being forced to do APRs because they couldn't get a 2-centimeter distal margin, which was our original plan. After chemoradiation, we think that a negative margin is all that you need. And there have been plenty of studies that have confirmed that. But the problem is that you cannot tell if there's a single cell island sitting where you make your staple line just because there's nothing on the mucosa. So we're going to live with the 2% forever, in my opinion, because I can't, in good conscience, tell people that they have to have an APR and a permanent colostomy just because I need 5 centimeters below a staple line in 98% of the time.
Thank you. That's very clear. In the trial, you conclude that pending clinical oncological outcomes, the findings do not support the use of laparoscopic resection in these patients. Was this unexpected or expected for you?
So we did not expect so to get into this a little deeper before I make my statement on that, we originally thought that there would be no difference, and we intentionally made all of the tumors within 12 centimeters of the anal verge. So that means that the majority of tumors were either in the mid or low rectal area, not in the upper rectal. Now, I don't mean to criticize wrongly, Jaap Bonger and the European group, they allowed upper rectal and rectosigmoid tumors and made their statement based on those. Andy and the Korean group and I decided that we really needed to know what the mid and low rectal tumors were going to do. So we insisted that that be 12 centimeter margin, or 12 centimeter site below 12 centimeters in the rectum. If you go into that, what you find out is that as you look deeper into the data, the majority of the problems occurred in the low rectum. We were not allowed by JAMA to say that it's wrong to use minimally invasive surgery in the low rectal cancers. But we were allowed to say, and actually encouraged to say, that we could not, in clear conscience, say that there was no problem with minimally invasive surgery with rectal cancer. So some way we had to hedge. So when we published our data, the outcomes data, the cancer outcomes data in the Annals of Surgery 5 years later, that was 3.5-year survival. There was no change in the overall survival. There was an increase in local recurrence in the low rectal cancer group. And now, I think if we get the data from both of our trials and combine them, that there will be a significant difference in the low rectal cancer with minimally invasive surgery, and we may need to change our statement.
Okay, and so sort of going further with this, do you think that the approach to rectal cancer surgery is evidence-based? And, you know, has your views been challenged by running this trial?
Well, my views were, I thought that we would be able to achieve technically the exact same outcome with a laparoscopic approach and an open approach. I really think that for me to do a low rectal cancer minimally invasively is probably inappropriate, and I will do a combined hybrid approach where I do a mobilization of the splenic flexure, mobilization and ligation of the IMA, and then do the pelvic dissection through a fan and steel open incision so that I can actually have a trained hand in the pelvis and know exactly where my margins and my planes are going and know where I'm putting my stapler across so that I feel like I'm using a minimally invasive approach for the patient's good, but I'm using an open approach for the cancer treatment portion.
Okay, thank you. And again, I'm very junior in the scheme of things, but I think sometimes the difference between the dissection and the transection is significant, where the transection's a lot easier to do open, which is not necessarily the same for the dissection. But I'm going to leave you with one last question, particularly given that you've mentioned collaborating for the meta-analysis with the other continental trials. What do you think about sort of colorectal surgeons trying to do more collaborative RCTs around the world? Is it feasible? Given the differences in the ways, arguably, these medical systems work, do you think it'll give us better results? Do you think it's going to be less relevant to any local body?
I think it's this is a global healthcare system now. It's not national. It's global. We have to be held responsible to our colleagues across the world. The way this happened was based on the tripartite meeting. It was terrific. Andy and his crew were there, and we went out on a tour of the bay, and we swapped protocols, and we talked about what we should do. And I have to say that if we hadn't done that, I would have felt really that we didn't have an answer, because it could have been just the American patients that we chose. I mean, obesity, narrow pelvis, we had a predominance of males in our group, et cetera, et cetera. Had Andy not done it in this with using the same protocol with a single variance of having 8% difference versus 6% difference, I think we would have had a mistake. We would have not gotten the right answer. So I would encourage the next generation of colorectal surgeons to really think about the collaborative effort that could be made that would benefit all of our patients throughout the world.
Thank you. I think that's a wonderful concluding statement. Thank you very much for your time, and thank you to the team in Dallas for hosting this event. I think it was fantastically run. I personally have learned quite a bit. And thanks everyone for joining in, and hopefully hello to those who are going to watch it on YouTube.